student in the Ecology and Evolutionary Biology department. When being at a university, I thought it would be appropriate to talk about how these tools we've been talking about all day can be used in an undergraduate classroom. And so I'm going to share some of my experiences in doing just that. So I'm in the ecology department, and I think this is the kind of thing that ecology conjures to most people. Beautiful landscapes full of diversity. I highly recommend you go to Antelope Valley if you haven't yet this year. This was last weekend. But as I got more and more into ecology research, I've started to realize that ecology research really excels when really cutting-edge quantitative and computational approaches are applied to compelling questions that come about based on observations in nature. And the thesis of my little talk here is that presenting quantitative and computationally challenging concepts using R and Shiny can really get undergraduate students who are into nature but maybe afraid of mathematics to start using those tools to ask their own questions about nature. To give you an insight about what kind of quantitative or computational tools I really mean can be used to study this sort of system, you can imagine me in the field asking questions like, how are these poppies and these white flowered cream cups competing with each other for the same nitrogen in the soil? And if I have that question, I can start writing a model in which the poppy population is one variable in a differential equation, and the cream cups, the white flowered plants, are another equation. And the populations of these two are growing at some rate that is reduced by the degree to which both plants compete with each other. Alternatively, I also study microbial ecology, and a question that I often have is, what the heck are the microbes in the soil doing now that these plants are growing above them? And also, how are they responding to all the rain that we got this year, which we haven't gotten for a few years now? And to do that, we can start collecting little samples of soil and using modern DNA sequencing, which gives us tons of data to identify what is actually in the soil. The challenge of this DNA sequencing approach, of course, is that it results in data tables that look like this thing on the bottom. This is just a snippet. These data files often have thousands and thousands of rows, each of which represents maybe one microbial taxon, one species, and each column represents one sample. This can be daunting for undergraduate students who really come into ecology thinking about beautiful plants or birds or butterflies. And so I'm going to present two packages or two little approaches I've taken to start breaking down this complexity in undergraduate classrooms. The first of them really leverages the DE-solve package, which is a differential equation solver in R that was built maybe more than a decade ago, I don't know exactly when. And the second is a package that I developed along with a group at UCLA called RANA-Kappa to start interactively analyzing those DNA sequence tables that I showed on the last slide. What I'm going to show here is just static images, unfortunately, from these Shiny apps, but there'll be links that you can go to if you want to play around with them for some reason. So the first is those Lotka volterra models, those population dynamics models I showed as differential equations, can really be turned into essentially little parameters that you can tweak around and start developing biological insight. For example, we can have students play with an app like this, where they can tweak around the parameters in that model, and for some parameter combination, they can see that both the green and the purple species coexist, and that the green is at a higher population level than the purple. But if they tweak that parameter combination, maybe the green one becomes more abundant and the purple goes away. Whereas if they tweak a different parameter to be higher, the purple one now dominates. So again, this is breaking down those complex looking differential equations into little things that students can interact with and interpret in terms of the actual nature that they came into an ecology classroom to study. Now to switch into this other Shiny app. This app was really designed to take tables that look like the one on top and an associated metadata file, which I'm not showing here, and to start helping students kind of visualize what this means in nature. This app, for example, it's online, and you can just play with a demo data set. And the demo data set was collected in the Pacific Ocean. And so these are all, each bar is a sample, and each of those things within each bar is one species. In this case, I'm highlighting the wrasse. And students can hover over each little color segment of that bar plot and see what species is present in which site in the Pacific Ocean. Alternatively, they can kind of group things and say, hey, the water next to Santa Barbara Island was maybe more diverse than the water next to San Miguel Island. So again, just to summarize, there's a lot of quantitative and computational concepts in ecology which I think can be made easier for undergraduate students to study with Shiny and R.